Okay. Now we're recording.
Okay.
And I will share my screen to present the speaker again.
So should I stop sharing?
I think actually all people can see out my screen without you stop sharing. Oh, I can do. Yeah, in this way it's fine. Okay, so thank you for being here to the Population Dynamics virtual seminar. So today we have Daniel Rodriguez Amor from the École Normale Supérieure in Paris. And the title of his talk is Cooperation Driven Multistability in Synthetic Microbial Communities. A few words regarding Daniel. So he got a PhD at the University of Girona, Spain. Then he got a postdoc at the lab of Ricard Soler in Barcelona. And he moved at MIT with Jeff Gore to work as a postdoc again, and then he became senior postdoc at the University of Graz, and finally junior professor at the Department of Physics, École Normale Supérieure, where he's actually now in Paris. And he's interested in several topics regarding how microbial interaction drive the dynamics of microbial communities and their functions. Also in ecology, how ecological regimes impact the spread of antibiotic resistance in community context, and is exploring these topics by using theoretical modeling and lab experiment together. So with this one, I would like to leave the stage to Daniel. Thanks for being here, and you can actually start. Okay, I think your screen is already there, and you can go on with your talk. Thank you.
Okay. Thank you, Marco. Thank you for the invitation. I'm very glad to be sharing with you some of our work. I'm going to be talking about the ecology of microbial communities. As you know well, in microbial communities, there are typically many different kinds of microbes that they interact in different ways. So some of them can be cross-feeding, so meaning that the secretions of some microbes are used by other microbes to sustain growth. In this way, it's a kind of cooperative interaction. So it can be cross-protection, another cooperative interaction that can protect one species from antibiotics because of the action from another species who might be actually secreting enzymes that deactivate the antibiotic. Microbes can be much more aggressive, secreting toxins to kill their competitors, and of course, they are all living together in an environment with limiting resources, so they can also be competing for those resources. And all of these interactions influence not only the stability and dynamics of the microbial communities, but also of the ecosystem where they live. And for just to show one example of how they might influence these dynamics, I'm showing here the results of a study in which the scientists were monitoring the state of the microbial community in the gut of a human subject. And we see that for many weeks for many weeks at the beginning of this monitoring, we have different groups of microbiota that are these lines in different colors. So each line is a group of microbes that have similar dynamics. And we see that the blue group here is dominating the system for several weeks. And then one day, this individual got an infection. It was a salmonella infection. And we see this group in green that go up and down in a process that took more or less about 2 weeks for the infection to kick in and then for the subject to heal. But during this time in which the pathogen was there, something changed in the competition between the other groups of microbes as well. So that once the individual heals, the state, the community seems to be in a very different state from the one that it was before. So now we have the orange group of microbes dominating the system, and others are at a lower relative abundance. So just to give you an outline of my talk today, I'm going to be mostly presenting past work which I did with several colleagues at the Gore lab. And those works were trying to understand better the responses of microbial communities in the lab, the responses to perturbations, and also how, as we increase the complexity of the community, we can have not only different responses to perturbations, but also a change in the alternative stable states that the system can exhibit. So in the main part of the talk, I will be explaining how different kinds of cooperative interactions influence these alternative stable states. And at the very end, I will briefly mention what we are working now currently in our group, which is all work in progress. So I will be happy to discuss as well about any questions you may have. So I'm going to begin with the first simple minimal community that we studied. This was while I was at Jeff Ward's lab. And his work I did together with my colleague Christoph Ratzke, who is leading his group now in Germany. So we start with these two bacteria that compete with each other, and this interaction is quite strong. And we knew that the mechanism driving this antagonistic interaction is actually how these microbes interact with the pH. So we have that the Corynebacterium has a metabolism that increases the pH. So when we culture it in monoculture, the pH goes up over several cycles of the culture, and then it stays up. And this is actually beneficial for the growth of the green microbe. The orange microbe, which is a lactobacillus, has the opposite trend. So it's acidifying the environment, and this also benefits its own growth. So what we wanted is to make a community out of that and study its dynamics. So we do these daily dilution experiments in which we start with a low number of cells in the test tube, and we let them grow until they reach saturation, and then we take a sample and dilute it into a new test tube. And in this way, through different cycles, microbes can continue growing and interacting, eventually reaching some sort of steady state. And when we did this for these two microbes that strongly compete with each other, we found that there was a threshold in the initial relative abundance that will determine the outcome of this competition. So if we start with the orange microbe at a relative abundance above this threshold, this microbe will rapidly take over the system and win the competition. So the effective interaction is that the orange microbe is inhibiting the growth of the green microbe. And vice versa. So if we start with a low relative abundance of the orange microbe, then this microbe is going to lose the competition against the green microbe. And with that, we had, to show that these were actually stable states, we repeated the experiment in the presence of some immigration or dispersal. This means that every day when we are diluting into the community into the new test tube for the next day, we are also introducing some fresh cells of both species at the same abundance, but in low numbers, right? And even if we do this, this doesn't change the outcome of the competition, which shows that those states are actually stable. And with this, if we make this physical analogy with a potential landscape, what we have is a community that has two alternative stable states, and now we can apply different perturbations to understand how we can jump from one state to the other. One such perturbation that we were interested about was to study how they react to invasions by a third species. And we tried with different invaders having different outcomes, but we found this particular one quite interesting, in which we started our experiment with the orange microbe at a high relative abundance. So we are in this state at the moment in which we introduce the invader, and we have here this Pseudomonas chlorographis that enters the system. And at this point, we see that it can invade, so it goes up in abundance, but also the green microbe that was below the detection limit also starts to increase. So eventually what we have is some sort of temporary or transit invasion of Pseudomonas, because just 2 days later, it starts going down towards extinction. But this passage of the microbe influenced the competition between the resident two species that we had. So we have a transition towards the stable state in which now the green one is winning. And this actually reminds us the example I was giving in the introduction, in which in a much more complex system, there was the passage of one microbe, in this case the pathogen, had changed the state of the community. But here, the nice thing about having this minimal system is that we can look into the mechanism to understand how these transitions happen. And as I introduced at the beginning, we have one microbe, the orange one, that it's decreasing the pH. So it means that it's increasing the proton concentration in the system. And this is beneficial also for the growth of that microbe. And the opposite is true for the green microbe. What we found when studying the growth of the invader is that it had a different interaction with the pH. So it actually grows well in acidic condition, but when it grows, it's changing the pH towards higher levels. And actually, during the experiment of the invasion, we see this transition in the pH right after we introduce the invader. The pH goes up, and the moment it's already at high pH, then the best competitors ends up being the green microbe, and the invader loses the competition against it. So we could also capture these dynamics in different models, including this mechanistic interaction with the pH, but we can also, depending on what our goals are, we can coarse grain a little bit and just capture these kinds of interactions with a simple Lot-Cabot-Terra model. So in this model, we have the typical growth for, let's say, our two species, the first species and the second species, that grow logistically. So they have this self-inhibition, so that the growth cannot continue forever. And then we had also this inhibition by the competitor. So this term captures how strong is the competitive interaction by the presence of the second species. And in order to capture as well the situation in our experiment, we have some dispersal. This term is for the fresh cells that we are introducing into the system every day. So just by introducing a third species and looking for different parameter values, we could capture these potential invaders that induce a transition between alternative stable states. And we also study different perturbations, such as antibiotic exposure that has straightforward interest in the clinics. And what we found is that when we introduce a modification in the model to account for the impact of the antibiotics, which here we capture as a death rate, we saw that whether you can use an antibiotic to induce a transition from one state to the other state is not so straightforward as looking at the death rates, but looking at the impact of the antibiotic to each species. But actually, there's an interplay between how susceptible a species is and how fast it can grow once you remove the antibiotics. So we could understand what we were observing in our experiments because of this modeling. And then we tried with different antibiotics and different growth rates of the species to verify that there is this actual interplay between these two variables that are important. So with this, we are in a situation in which for the simple model with two species, we can understand relatively well how a bistable community responds to different perturbations. And now I'm moving to how these alternative stable states in complex communities may emerge, not only out of interspecies interactions, but also on different modes of growth or intraspecific interactions for our species. So this is work that Will Lopez, a postdoc at Jeff Kor's lab, did very recently, and he was interested in making an experimental system that was representative of the human microbiota. So he selected these 6 different species that come from nasal isolates. So this means from the nasal microbiota. And then he started by just studying what is the dynamics if you start the community from different from mixing all of the species at the same initial ratio, and then do 5 of these daily cycles of daily dilutions. And then what he saw is that we observed that the different replicates were reaching different outcomes. So for example, we have 2 of them that at the end of the experiment were dominated by the yellow microbe. And then we have also another 2 of them in which the blue and the red microbe were still competing. And we have 2 other replicates that went to a different state in which only blue was present in the community. So when we saw this, we thought that perhaps these different outcomes of the community were representing alternative stable states in which the community could fall. And to understand this hypothesis, what Will did was to repeat this experiment, but now the initial condition is such that 90% of the population comes from just one species, and then we try with the rest of 5% divided among the other species. And when he did that, we saw that the community actually falls into 4 alternative stable states. So when we start with 95% of the blue species, we have that SE, so this Staphylococcus was taking over the community. But if we started with 95% of the yellow one, then it was this one, Staphylococcus aureus was dominating the system at the end of the experiment. Same with Moraxella in green and with Cryptococcus in red. Okay, and then we have 2 other species that seem to be losers of the competition, because even if they started at 95% of the competition, by the end they were defeated, and Cryptococcus was taking over the community. So here we have 6 species, different alternative stable states, and we wonder whether the interactions between any 2 species could explain what happens to the complex community. And for that, Will did these experiments in which he competed each species with another one. And we found that among the four focal species that were taking over the stable states, we saw bistability in all of the possible combinations. So basically, this means that SE can win against any other of the competitors, depending only on the relative abundance at the beginning of the experiment. And this is true for all the combinations. So this was quite surprising because just from an intuitive perspective or coming back to even the modeling, we know that there are there are three different outcomes that can happen through the competition. So depending on how strong is the inhibition by the competitor, we can have generally these three canonical outcomes of the competition. So if the interaction terms or the competition between the species is not very strong, we will expect some coexistence among two pairs of microbes. If one of the interactions is strong but the other one is relatively weak, then we could have cases of exclusion when only one of the microbes win. And only in cases in which the interactions between the two microbes are very strong, then it's when we can have bistability. So out of the previous experiments in which we found bistability everywhere, if we think that everything is driven by the interactions between the species, this will have meant that all of the interactions between them are very strong. And Will was quite not convinced yet. So what he did is to try to assess interactions in a different way. And for that, he turned into this spent media experiment. So basically, you are growing the one microbe, and at the end of the cycle, you remove of the cells, you take the supernatant, and then you inoculate cells from another species, and you see how they grow. And this is what he found in different combinations. I'm gonna explain what this matrix is. So if you see red, it means that there is a strong inhibition or competition. And if you see green, it means that we have cooperative interactions. So basically, if we take the first square, we have CN growing on its own metabolic byproducts, and this seems to be inhibiting the growth of CN. So CN grows less in its own secreted medium than in fresh medium. But this is not true, for example, in the second row, in the case of Moraxella. Moraxella, if growing in the secretions of any other microbe, including itself, grows better than if it grows in fresh medium. Okay? So we saw different cooperative interactions out of this experiment, but also three cases of self-facilitation, which means that the species is also modifying the environment in a way that promotes its own growth. And what does this mean? So if we think about the typical logistic growth, which will have given you a red square in the previous matrix, what we have is a monotonically decreasing function of the per capita growth rate with the population density. Okay, but if we have cooperative interactions, what we have is that the maximum growth rate is not going to be at low densities, but at some intermediate level. And this is what we call the alley effect. And it can be classified into two kinds of alley effect. The weak one, which is showing this tendency, but at when the population is very small, there is still a positive growth rate. And the strong allele effect means that there is a minimum population size that is required for growth. If the population is too small, the actual growth rate is negative, so it means that it will go towards extinction. So based on the previous experiment and this interpretation, we'll check how is the growth of the microbes starting at different initial densities. And we actually saw that for CN, it was consistent with logistic growth, but for the other microbes, it was consistent with a cooperative growth. In particular, Moraxella was having a strong allele effect, and it required a relatively high number of cells in order to grow. Then we wonder what happened if we have these kind of interactions and intraspecific interactions introduced into the model where we have multiple species competing. And what I'm showing here is the phase space for the growth of CN species against the SE species. And in this case, what we see are in the dashed lines are the null lines, so it means along these lines, either the blue species or the red species do not grow. And then these arrows that you see are the trajectories. So if you start a community at this initial number of cells, where does it go? It goes towards the blue species winning. So here we have this value of 1 as the carrying capacity of the blue species, and 0 will be the final density of the red species. This will be if both of them are in the Lotka-Volterra model, in which we don't have cooperative growth. But if we introduce this cooperative growth or this allele effect, what we find is that the null line for one of these species bends, and now it enables the red bacteria that before was losing the competition. It enables that it can win depending on where we start the competition or what are the initial conditions of the experiment. Here we see that these arrows can lead us to either the red one winning or the blue one winning. So this was still our hypothesis, and if we wanted to test it would be nice if we could play with the strength of this cooperative growth, right? And it was not obvious to us how to do it, but Will went back to the experiment and started to make a huge bioassay in which he tried, I think it was 24 different molecules that could perhaps influence the growth of these microbes. And he actually found that glutamate was very important. So if we grew the Cryptococcus, so this red species, with added glutamate, its growth didn't change much compared to the original condition. But when we grow the other microbes, the ones that were cooperatively growing, what we'll saw is that for these yellow and blue species, both of them were improving quite much the growth rate at low densities, so somehow making it closer to logistic growth. So somehow it eliminated the cooperativity in their growth. And for Moraxella, it was alleviating the effects of this cooperation by just making the minimal population number required to growth, making it quite smaller. So this was great because now we can replay these competitions by eliminating this cooperativity in 3 of the species. And according to the model, there were many different things that could happen when you change from cooperative growth, so from this allele effect, where we have a lot of biostability, to something with a reduced cooperation or reduced allele effect, in which you can have more cases in which one species win. And actually, these map well into the experimental results for the competitions between these different species. But also there were many other things that could happen, like a case of tri-stability, so three alternative stable states in the presence of cooperation will change into coexistence, which means that here the blue and the and the red species could coexist. Actually, there's it should be between the two streptococcus. So just to summarize what happened in the absence of glutamate, we have a lot of cooperative growth, and this led to many cases of bistability, so between all the pairs of these four species. But when we have reduced this cooperativity or the allele effect by adding glutamate, then there was only two alternative stable states. Either SE and SA were coexisting, or MC and SA were coexisting as well. The rest was at the expenses of the Cryptococcus losing the competition between any other. And again, this is what happened when we started the competition between the six initial species, and now we see that we end always in one of these two alternative stable states that I was mentioning. So up to here, I showed that we can understand pretty well the responses of antibiotics and other kinds of perturbations in minimal communities. And these responses are driven mostly by the interactions between the microbes and also between the microbes and their environment. For more complex communities, what we saw is that cooperative growth can lead to emergent alternative stable states. And here's what I wanted to present today about past work. And very briefly, I'm going to mention in which directions we are going. So basically, what we are now interested in to understand how more complex communities respond to perturbations. And very briefly, just mentioning that in a different work, we had also characterized the expected dynamics based on a statistical approach to microbial communities under competition, in which we have this lot-coupled terra model, and we are interested in the competition in a focal community. So we have, as before, the growth and the interactions, but now for many species. And again, we have a dispersal term, like in our experiments. But these dispersal models, the fact that in natural ecosystems, you often have immigration from a species pool or from a reservoir that are neighboring ecosystems, if you want. We are focusing on this focal community, and what we knew from the theory is that if we start a community with a low number of species, and we sample the interactions between the species from a given distribution with a given average. Here we have this average of 0.3. So if we don't have many species, this is the panel on the left, all of these species tend to coexist in the ecosystem in the long term. And now we play the game of increasing the number of species. What we see in the second panel is the case where we have 20 species, and in this case, many of the species will still stable coexist after some time, but some others go to extinction. They don't go fully to zero because we have this dispersal term. But if we stop the dispersal and we set it to zero, all of these that are below the dash line will go extinct. And finally, we can keep increasing the number of species, and we see a transition to a persistent fluctuation dynamics. This is more chaotic, and here in this regime, we have typically the rise of a given microbe, but it has kind of its moment of glory, but then the next day it's going to be replaced by a different microbe. And this situation in which species are going up and down in abundance keeps on in the long term. So the same kind of phenomenon happens not only if we increase the number of the species for a given distribution of the interaction terms, but we can also fix the number of the species. Here are 50 species, and we increase the number. We increase the average interaction that we have in our distribution where we sample the interactions, and we have again the same three phases. And we actually verified it. Gillian, who a PhD student in the lab at that time at Jeff Gore lab, did this experiment where we actually quantified and validate these different dynamics in experiments. So with this and the previous work that I discussed today, we lay our foundations to go into the responses of more complex communities. So in these last 2 years, I built a team of an interdisciplinary team of scientists with different backgrounds, going from theoretical physics to microbiology. And I'm gonna tell you about the experiment that Sarah Timsit, a student that did the master thesis with us, and Stella were doing over the last months. So here we have communities with different species. I'm showing you the case of a 3-species community, and we see the time series for the total biomass, which we measure through the OD, over several days. And then at day 6, you see this red rectangle. At day 6, we apply an antibiotic shock, and we see the total biomass going down. Then we remove it, the antibiotics, and we see that the biomass can progressively recover. But we can do that modeling what will be kind of an infection experimentally. This means that we introduce an invader before the antibiotic treatment. So here, this invader is a species that was isolated from a patient in the hospital. So it's a pathogenic strain of E. coli. We introduce it in the community on day 5, and then after it arrives in the community, that's when we have an antibiotic treatment. And we see that when this pathogen or this invader is susceptible to antibiotics, we can see that the total biomass goes down and then it recovers. And if we introduce a resistant pathogen, then the total biomass during the antibiotic treatment doesn't change too much. This seems quite intuitive, but it doesn't need to be like that, and it's not always like that. Actually, here are the results for another set of species. We have a community with 28 different species. 18 different species. And actually, it seems to benefit, at least in terms of the total biomass, after we put the antibiotics in. And the dynamics that follow after this depends also on the different invaders that can arrive into the community before the treatment. So right now we are trying to understand what kind of communities will react in a way or another. We just had the sequencing results back a few weeks ago, and we are still analyzing them for this kind of experiment. And we are also obviously looking for statistical patterns that can help us make sense of which communities might be more or less resilient to the antibiotic treatment, for example. And this is something that before having these sequencing results,
Sorry, Daniel, we have a
yes,
question from the chat, and
yeah.
this Poina is asking whether these experiments are done in batch culture, the one that you just showed.
No, these are daily dilutions. So it depends on what do you mean by batch culture, but I understand batch culture as just let them grow and stay. And this is not what we did. So here we are still doing the daily dilutions every day, right? So we start with these 18 species at day 0. We do 5 daily dilutions, and then on the 5th dilution we introduce the pathogen or this invader, and then the next day we apply an antibiotic treatment.
Yeah, thanks.
Okay. So then Jorge took the data of all the communities. We have about 800 communities with different kinds of conditions, like either we introduce the invader or not. It could be resistant or not, and whether we had no antibiotics or 1 out of 2 antibiotics that we tested. And just training a simple neural network with the data of the biomass until day 7 and the identity of the initial species, including also these invaders. The model can predict the biomass after the treatment, right after the treatment, for all the for all the samples that were left out of the training. So here we are at each time leaving one out and trying to predict what will be the response. So somehow we can capture just in this initial set of species, we can capture some patterns or that might drive how they react to antibiotics. So we want to combine different tools also with the sequencing data that we just got to try to understand a bit better how complex communities might react to perturbations. And with this, I would like to thank our group and also thank the people that contributed to the work that I presented. And I'm happy to take questions.
Thank you, Daniel, for a very nice talk. So I will